And by this hybridization, then they mark the mRNA for degradation by an RNase. There is one approved antisense therapy drug. It's an antiviral drug. It's not a big seller, but there has been one that has been FDA approved, and there are many more under development at several different companies. The really exciting new drug, for which there's been just incredible capital investment in the States, takes advantage of this process of RNA interference. And I'm just going to sort of point out here, I guess, that this is an RNA-based drug that has in common the ability to target and degrade a specific mRNA sequence. And so it has that in common with antisense therapy approaches. These companies, such as Alnylam, which is one of the biggest ones that was formed initially around this biological phenomenon, it widely recognized from the beginning that delivery is going to be a big issue with RNA. And again, there are methods for chemically modifying RNA, but in this case, it's still a little uncertain what types of modification and how much modification you can get away with, because there is a very specific biological system that these drugs have to utilize to affect mRNA degradation. And it's still a little unclear how modifications of the siRNA are going to affect that process. Now, it has been shown, though, that stabilizing nucleic acid modifications can still allow function, but it's still relatively early. The approaches are basically the same for shorter DNA segments, oligonucleotides, for antisense, and for silencing RNA. There's the same cationic polymer approach to condensing the nucleic acid, smaller nucleic acid drugs, so you get polyplexes, and also cationic lipids for lipoplexes. And so the delivery technology looks very similar at this point from this general standpoint. In summary, then, I would say that early clinical experience with these non-viral delivery methods has been quite disappointing. Several of the companies that were formed around the new cationic lipid technology, as well as polyplex, so polymer-based technologies, have gone by the wayside and had disappointing clinical results. The basic reason is that biology is really hard to beat. Conservative estimates of the difference between analogous viral delivery methods, so say for the same plasmid or gene, versus non-viral, are better by 10 to the 6th. I've even seen 10 to the 13th demonstrated in the literature. I mean, that's a huge number that you're up against. And so at this point, a lot of people are sort of wondering, are we ever really going to have non-viral delivery methods that find their way to real-life products? The reasons, again, are because there's all these barriers, especially from a systemic delivery standpoint. When you're talking about these biomolecular drugs, there are huge issues of stability, transport, how they get picked up in various places. They have very poor pharmacokinetic properties. Targeting, how will they ever find their target cell and get inside? These have to get inside the cell, that's a key point. And so intracellular delivery has really been shown to be a huge barrier in vivo. Even if they get to the right target cell, getting a macromolecule or a nanoparticle inside the cell is far from trivial. Getting it to the right place inside the cell is what's really trivial. So I'd like to just close to give you an example from our own group of, I think despite having said all those negative things about what we find interesting, number one, it's a challenge that if you're sort of on the delivery side, you just can't get away from it. It's just such a big challenge. There's such a need for it. But where we got interested was actually from thinking about the good news. And the good news is you always have to remember that biology has figured out a way to do this. And so there is a design example out there that we can look at. And if you begin to look at it in more detail, you don't really think anymore that these viruses are simple organisms. They're amazingly complex and they're amazingly efficient. And what they really are built to do is to deliver macromolecules to the right place inside the cell. That's really what they do in an unparalleled way. And what probably is well appreciated, and Steve was talking about this, are some of the details by which they very efficiently enter the cell through receptor-mediated endocytosis. But what I think is way less appreciated is that many viruses and many pathogens, so protein-based pathogens such as diphtheria, cholera, share this common challenge of the fact that when they get taken up, they're still not inside the cell. They're in a membrane-bound compartment called the endosome. And the problem with this, if you're a macromolecule, biological macromolecule, is that this gets evolved into the lysosomal compartment where they are degraded, or they get recycled back outside the cell. And so what pathogens have evolved is a delivery system that's activated inside the endosome, and it's activated by the fact that these endosomes develop a low pH gradient. They go all the way down to pH 5 to 5.5. And they have a protein that converts at low pH to a membrane destabilizing form and activity. And so only once they're inside the cell, inside the endosome, is this protein activated to destabilize the membrane to aid the transport of their drug, be it DNA or protein, to the cytosol. This has been studied in some detail, for example, in the flu virus, that is schematically shown here. There's a protein called hemagglutinin that is activated. At pH 7.4, it's in a stealth-like conformation. It has no activity, membrane activity. But as the pH drops inside the endosome, so down here would be inside the endosome, as the pH drops, there is a triggering of a large conformational change in this protein, and it exposes side chains that partition into the endosomal membrane and destabilize it. And in this form, then this membrane destabilization allows the effective transport of these relatively large particles across that membrane into the cytosol. And it's been studied in some detail, but this is the question that we got interested in. Could we mimic these biological machines with safer synthetic systems? And so what we would be after is the synthetic analog to these viruses that can enter into cells efficiently. That part is fairly easy. But have this additional activity that synthetic systems really haven't had in as the pH drops in a reversible manner, the delivery system would destabilize the endosomal membrane and get the nucleic acid drug to the cytosol. In a little more detail, the mechanism of hemagglutinin is described even better. There is a very specific glutamic acid side chain with a carboxylate residue, and it's the simple protonation of that side chain that triggers this large conformational change in the protein. And it's because the pKa of the carboxylate is tuned to this pH gradient that occurs in the lysosome. The pKa can be tuned by the residues around it to that region around pH 5.5 that these pathogens find in the endosome. And so they go between a hydrophilic, stealth-like conformation to a hydrophobic alpha helix that is membrane destabilizing. And it's very important to recognize, because this is a protonation-deprotonation event, that this is reversible. And this is how the pathogens accomplish this without being particularly toxic, in many cases, to the cell. Once it goes back to the cytosolic pH, then it becomes this hydrophilic polymer again. So we were after a synthetic equivalent. I'm not going to go into a lot of the chemistry, but you can see that there's two key pieces to our design. There's a carboxylate residue that is the same pH sensing unit that's found in these pathogenic systems. But then we have alkyl segments, both within the same monomer, but also can be provided in different monomers. And it's that alkyl segment and the details of that are related to the membrane destabilizing activity. It's these groups that when this carboxylate becomes protonated, partition